Welcome to MS Research Australia's third Progress in MS Research live update. Thank you for joining us as we stream across Australia and around the world. I'm Dr. Hamish Campbell and I'm the Deputy Head of Research at MS Research Australia. I'm joined by Bryony Hinu, who is joining us as a co-host today. Bryony has been living with MS for the last 3 years, and actually she was one of our top 10 fundraisers for Kiss Goodbye to MS' latest campaign, the May 50K. So thank you very much for coming and joining us and being a co-host.
Not a problem. It's a real honour to be here sharing the stage with you, Hamish, so thank you for the invitation. I think that making this live stream event today available and access to stream for everyone, it's a really fantastic way to be able to connect people living with MS, such as myself, as well as our family and friends, to the latest research developments that are going on in MS. So thank you to MS Research Australia, you and your team, for bringing some of the top MS experts together from both Australia and internationally together today for this live update. Before we begin proceedings though, and on behalf of MS Research Australia, I just wanted to start by respectfully acknowledging the traditional owners of the land that we're meeting on today, which is the Wurundjeri Woi Wurrung people of the Kulin Nation, and pay respect to their elders past, present and emerging.
Excellent. Thank you for the acknowledgement of country, and I'd also like to thank our sponsors Roche for making this event possible, and also I'd like to thank the Florey Institute for Neuroscience and Mental Health for generously providing the venue from which we're streaming from today. Now, research is progressing at an amazing rate, and we're going to hear about some of the amazing research that's happening in Australia and also around the world, but we couldn't fund MS research without your help. Currently, less than 2% of our funding comes from government sources, so we rely on you to be able to fund this vital MS research. So make sure you go to our website, msra.org.au/donate, to really donate those vital funds that we actually need to fund some of this amazing research that's going on. I can't tell you how much we appreciate your support. We're going to hand back over to Bryony now, who will give you more insight into kind of what we've got in store for you today.
Wonderful. Thank you, Hamish. So let's run through what we're going to be covering on this morning's live update. So we are live streaming to you today, hot on the heels of MS Research Australia's conference, their latest Progress in MS Research Scientific Conference, which actually concluded yesterday after 2 jam-packed days. So Hamish is going to fill us in on the key research that was covered in those 2 days, and then after that we're going to speak to 4 different experts in the field of MS research on some really hot, interesting topics, including environmental risk factors, remyelination, self-reported outcomes, and the evolution of the Australian MS treatment landscape today. You should be seeing some more details about our 4 speakers today and their topics that should be showing on your screens. After that, we will have the opportunity to put your questions from you at home to the 4 experts that we have with us this morning. So thank you to all of those at home that have already actually sent in questions with their registration for today's event. We've gotten quite a few, which is fantastic, and there are some really good, interesting questions in there. The team at MS Research Australia have been putting in a lot of work collating those questions to answer them as best we can. If you haven't submitted a question but you're watching this morning and you do think of something that you'd like to be posed, there's still time. Simply send us a message either via the Facebook page for MS Research Australia, or you can drop an email to inquiries@msra.org.au, and the team are on their laptops ready to take those questions in. So without further ado, Hamish, can you give us the rundown on what happened at the scientific conference the last 2 days?
Yeah, absolutely, and I suppose I'll put it into a bit of context. So we run this conference every 2 years, and we invite all the kind of researchers in the MS field in Australia, and there can be some from other fields as well. And so we've had about 155 scientists here in Melbourne, and that has included neurologists, MS nurses, right through to kind of fundamental biological researchers, and also those in kind of the social and applied kind of space. And so it's been really good, and I think kind of one of the really kind of encouraging things is seeing some of the young researchers that are busy coming through and some of the excellent research that they're doing. I think kind of the field is in a good space, and I think there's a bright future up ahead. And I suppose the purpose of the conference is really to stimulate that innovative research into the causes of MS and also the prevention, but of course ultimately it's the cure for MS that we're kind of really working on. It's also also to promote the exchange of scientific ideas and resources, and kind of trying to make sure that kind of neurologists are talking to fundamental biologists so we can really address the questions that are vital in the field of MS research. So it's really kind of encouraging and supporting collaboration amongst kind of not only amongst Australians but also further afield as well. I suppose some of the key themes that have emerged, one of them I suppose was kind of looking at ways that we can do clinical trials better, hopefully maybe a little bit quicker, because it's very important that we kind of get new treatments into the clinic as soon as possible. And it's not only how we carry them out, but are there better measures that we can use to kind of dictate success of a clinical trial. And this is particularly important for progressive MS. It's a great unmet need in the MS field. We've got a number of treatments now for relapsing remitting, but we're still very short of treatments for progressive MS. So one of the hurdles to that is actually working out ways we can actually detect progression in a clinical trial. So that's trying to kind of detect kind of progression in a kind of shorter kind of time frame, I suppose, is one way to look at it. And so scientists in Australia and around the world are looking at things called biomarkers, which is short for biological markers. So these are something that we can readily access and can reliably test, which they may not be causing the disease itself, but it's kind of a measure of really what's happening under the hood of MS. And so there was a number of different talks at this conference talking about that, and they include things like looking at genetic material that's floating around in someone's blood. There's also a kind of a novel use of MRI to really kind of pin down and really look at kind of individual axons in the brain. And the technology has come along such a long way, and that was really fascinating. And also people are looking at kind of wearables, so things like Fitbits and things like that, to kind of help try and kind of build a full picture of someone's MS. And these studies, as I said, are very important for progressive MS and for clinical trials, but it's also very important for relapsing remitting MS, so we can hopefully kind of have more rapid changes of medications should someone's medication not be ideal. So I think that was actually really interesting. But of course, if we're going to have clinical trials, we've got to kind of also have kind of treatments to kind of feed into these clinical trials. So it was also very interesting to hear some of the kind of potential therapies that are coming through. We heard it from a group of scientists who are working on copper and kind of redistributing it around the cells of the body, and I found that kind of very fascinating. And also scientists are kind of using cells as therapy, so kind of trying to use cells as a medicine to kind of target kind of certain parts of the body. And so I think that was very interesting. We also heard about kind of remyelination. So in MS, the immune system attacks the protective coating on nerve cells, and so ways that looking to kind of protect that. I won't go into too much detail about that because one of our international speakers is an expert in that area, and I'm sure he's going to explain a lot more about that. But that was also very interesting. And so I suppose another very important aspect was kind of using patient-reported outcomes, or patient-centred care as we refer to it. And this is kind of trying to include patients and their feelings about their MS, because no one knows someone's MS better than the person themselves. So it's trying to look at ways to kind of include that as a measure and kind of objectively measure that and include it with kind of the more traditional methods of MS progression, so we can really kind of get a full picture, once again, to help drive therapeutic decisions and also clinical trials. So I think it was very, very interesting. I probably should leave it there because otherwise I could probably go on forever. But I will just say there was a whole range of things. There was also looking at diet and the risk of MS, and also things like electrical stimulation of muscle for rehabs and things like that. So it was very interesting, but I'll pass it back to you.
Wow, there's a lot that's going on, clearly, and a lot of people involved to be covering all those sorts of different aspects of research, so that's fabulous. So look, before we do meet our first researcher this morning, don't forget that you at home, you can get involved in the conversation today through social media. You can just simply head on to Facebook or Twitter, and we do have a hashtag. It is hashtag MSLiveUpdate19. So please do get involved. If you have any feedback or questions, you can fire away with that hashtag on those social media channels. So it is time for us to hear from our MS experts on some of those very interesting topics that have been covered in the last couple of days. And as a person living with MS, I'm certainly very interested in hearing about them. So
yeah, let's get started. So all the way from the United States, we have Associate Professor Alan Mowry, who is from the Johns Hopkins University. Associate Professor Mowry focuses on environmental factors that influence the risk and progression of MS, as well as improving outcome measures for the use in clinical trials, and looking whether high-dose vitamin D reduces the risk of MS activity. She's also working on redefining personalised therapies for people with MS, and is assisting colleagues with study design and overseeing how MS clinical trials are conducted. So thank you very much, Alan, for joining us. And today we're going to be hearing about deciphering environmental enigmas, MS risk factors explained. So welcome.
Thank you, Hamish. I'm so happy to be here.
So I'm going to fire off the questions. I hope you're ready. The first one, environmental factors are known to be important in the risk of developing MS. What are some of the environmental risk factors associated with MS?
Yeah, that's a great question. Recent international genetics consortia, including many investigators here in Australia, have demonstrated now that there are more than 200 gene variants linked with an increased risk of MS. However, they really explain a fairly small proportion of why people get MS. For an example, if you had an identical twin and you were living with MS, her chance of getting the disease would only be about 30%. So a lot of MS risk is probably related to environmental factors. There have been several factors identified to date. Some of them are things like Epstein-Barr virus infection, and then cigarette smoking has been known for quite some time to influence the risk of MS. Other things include vitamin D status, people with low vitamin D potentially having an increased risk of MS, although that story is becoming increasingly complicated. And the most recent, perhaps identified risk factor is obesity, particularly in late adolescence.
So if we can just go back to the vitamin D, you're saying the story is getting a little bit more complex. Can you explain that a little bit?
Absolutely. So the earliest studies looking at vitamin D status and MS risk were born out of the observation, one that's actually been established again with a study that was presented over this conference, that MS is actually more common the farther away from the equator we live. And so this notion of something related to sun exposure being the causal factor in that interesting observation came about. And of course, most of us get our vitamin D from the sun, so it was a logical thing to study. There were a few studies in which people in different parts of the world had banked blood prior to developing MS, and so the investigators looked at those blood banks, measuring the vitamin D levels in the blood in people who later went on to develop MS compared to those who never did develop MS, and showed, yes, even 5-10 years prior to developing MS, vitamin D levels were lower in those who were going to get the disease compared to those who never did. So it seemed like a really compelling story. However, recent data, including by Professor Robin Lucas at the ANU, have demonstrated that the story is a bit more complicated, and that maybe something about sun exposure itself, possibly even UV light itself, which is known to impact the immune system in ways other than just by increasing vitamin D levels, that seems to be even a stronger association. So I think that the story gets a little bit muddied, actually.
And at our conference, I think the story even got a little bit more kind of complex, because we heard about kind of genetic variants that people have, which actually affects the way they metabolize vitamin D. So that's another layer of complexity on top of that.
Absolutely.
So gut health and microbiomes and diet have been associated with the risk of developing MS. So what can you tell us, and what do you know about diet and microbiome in MS?
Yeah, this is a really exciting area, and one that people with MS have been asking us to look at for quite some time, so it's great that we've arrived. The microbiome, in this case, we often refer to as the gut microbiome, or the bacteria that make up the composition of our gut. And this is a very interesting topic, because we know that our immune systems are educated to a large extent by interacting with those gut bacteria. And so there's a hypothesis that alterations in the bacteria, or how the immune system is interacting with those bacteria at the level of the gut, may underlie a number of autoimmune diseases, including MS. And then, of course, diet can interact with and contribute greatly to the composition of those bacteria. Some studies have already demonstrated from the perspective of microbiome that there are differences in the microbiome of people living with MS compared to those without MS. However, those studies are a little bit tricky because by the time a person is living with MS and can have their gut microbiome studied as such, there may be changes in the motility of the gut or medications that are used to treat MS that can affect gut motility that may cause a shift in the microbiome. So it's a pretty complicated area to study. With respect to diet, we've heard some of the hot off the press research from Professor Lucinda Black that really provided the first comprehensive investigation demonstrating fairly strong association between dietary composition as a whole and MS risk at this conference. So what she demonstrated is that people who eat a Mediterranean-style diet with a little bit of red meat added back into the diet actually had a much lower risk of developing MS. So those data were really exciting. And then finally, she and others have worked to evaluate the impact of fatty fish intake on MS risk. And 2 studies now demonstrating that people who eat more fatty fish have a lower risk of developing MS. So some really exciting recent work, again, led by many of your colleagues here in Australia.
And you're doing your own research into this area. What can you tell us about that?
Yeah, so we were very excited to think about how obesity, a known risk factor for developing MS, may impact the prognosis of those who've already developed the disease, whether we can modulate that. And we have some interesting work on the microbiome as well. So our first study really looked at obesity and the impact on the health of the brain overall. And what we showed in a large group of individuals living with MS is that those who were obese, particularly morbidly obese, had an accelerated rate of shrinkage of the brain. This is really relevant because we know that brain shrinkage is an increased risk factor for long-term progression of MS. And to that end, we really wanted to focus on whether diets that may reduce the risk of obesity could be impactful on the disease course. So to date, we've done a small pilot trial doing a fasting-style diet where people ate a very low number of calories on certain days. What we found is that people who were enrolled in the study and who did these calorie reduction diets lost weight, and as such they actually had clinically meaningful improvements in their depression, which already is a win for people living with MS. We also saw that the weight loss was associated with an improvement in the diversity of the gut flora, as well as with biological change in what we call the metabolome, so kind of evidence of how the body systems are working, that are quite relevant to both the nervous system's health as well as that of the immune system. And then our metabolism studies have also demonstrated that people with MS, particularly with progressive MS, have lower level of gut-derived metabolites than individuals who are otherwise healthy. And to that end, in particular, we are studying bile acids, and we are studying whether supplementing people with these bile acids leads to any meaningful improvements in outcomes, particularly in progressive MS.
Yeah, that's really interesting, and I suppose a little bit surprising. I suppose you weren't necessarily expecting that. I suppose that's all we have time for, so I'd like to thank you very much for coming and talking to us, and we wish you all the best in your research, which is ongoing.
Thank you,
So thank you very much. Before we move on to hear from our next MS expert, it's time, if you haven't had time to submit a question, now's your time to do it. You can send us an email via the email address which is on the website, inquiries@msra.org.au, or you can submit a question via Facebook. While we can't answer every question due to time constraints, our research team are working hard behind the scenes to compile all your questions, and those which we aren't able to answer live, we will respond to you as soon as we possibly can. Remember, if there's someone who you think would like to watch this stream, remember you can share it on Facebook or you can share it via our website. Also, don't forget to join the conversation on social media using the hashtag MSLiveUpdate19. Now back to to you, Bryony. What have we got coming up for us?
Thanks, Hamish. So next up, we're going to hear some information about repairing MS damage or remyelination research. And we've got an expert in this field. This is Associate Professor Gonçalo Castelo Branco. He's actually travelled to us all the way from Sweden, where he works at the Karolinska Institute in Stockholm. His research there focuses on what defines the epigenetic state of myelin-producing cells, or kind of in layman's terms, it's more the set of instructions that the cell uses to read DNA to then perform certain cellular actions. And so the aim of his research is to be able to eventually design therapies to induce myelin repair in demyelinating diseases such as multiple sclerosis. So a big welcome to you, Associate Professor Castelo Branco.
Thank you very much. It's a pleasure to be here.
No, it's great to have you. So look, I thought we might as well start with just the basic question of what is myelin, and why is it that it's relevant to multiple sclerosis?
So myelin is a sheath that insulates the nerve cells. And the nerve cells in our brain and in our spinal cord, they communicate with each other with electrical impulses. Now we can do an analogy between our nerve cells and the electrical cables that we have in our homes. So the electrical information in the electrical cables, to be transmitted fast, it has to be insulated. That's why we have those plastic surfaces. So the myelin serves the same purpose in our brains and spinal cord, and that allows a fast transmission of electrical impulses. Now, in multiple sclerosis, what it happens is that the body recognizes the myelin as non-self, and the immune system will then destroy and target the myelin. And this will lead that the electrical information is transmitted in much slower rate. Apart from that, the myelin also protects the nerve cells, and eventually, if you don't have the myelin, the nerve cells can die, and then lead to the symptoms that usually are associated with the disease.
MS. Okay. Yes. So it's kind of that insulation is broken, so it impairs your nerve function.
Yes.
So if that's the myelin damage, so what is it about remyelination? How would that be important in MS?
Yeah, so remyelination is the process by which the body repairs the myelin. So in our adult brain and spinal cord, we have progenitor cells of the oligodendrocytes, which are the myelin-producing cells. And in, for instance, relapsing-remitting MS, these progenitor cells can repair the myelin, and thereby we have some recovery of the symptoms.
Okay, and so the hot topic, of course, is the potential of remyelination therapy. So what could remyelination therapies mean for people with MS?
Yes, so remyelination process then would be able then to repair the myelin, and then there will be some restore the flow of electrical information, and then thereby tackle some of the symptoms of the disease. This is why it's a very hot topic. There is a lot of both fundamental science and also clinical researchers focusing on remyelination and new strategies to induce remyelination.
Okay, so it seems like it's an exciting new idea, a new kind of path that treatment could take for people with MS.
Yeah.
How far into the future do you think we're looking for remyelination?
Yes, I think it's looking promising. There are several clinical trials ongoing, some more advanced than others, some that have already some promising results, some of them that had some results that were not so promising, but they have now they are being redesigned in order to be able to tackle the question better. There is a lot of research also to try to find new targets that can be used for remyelination. We need to understand better the biology in order to understand how to tackle the remyelination.
Okay, so there's a few different directions that they're kind of approaching this research, and it sounds like there's a lot of people involved.
Exactly.
What kind of progress has your team, your research, been making into this area?
So my team, we are focusing in the fundamental aspect of research, and we are focusing on the molecular properties of these myelin-producing cells, the oligodendrocytes, and also their progenitor cells. And we have been using a new technique called single-cell RNA sequencing. sequencing. So when you try to analyze the brain, what happens before, you could not really go into the detail into the individual cells in the brain. So we can do also another analogy is like a smoothie, that like when you taste a smoothie, you have a taste of the different fruits, like the apple, the pineapple, the strawberry, but you cannot really know in detail. This new technology that arose in the last 10 years allows us to really go into the detail, into the different components, different cells of the brain and the spinal cord, which will allow us also to know much better what they are doing. So, for instance, what my team has been doing was that we have been able to see that the oligodendrocytes that produce myelin are much more complex than we previously thought, and we also have seen that in the process of in models of multiple sclerosis, we can actually see that the progenitor cells, apart from their function to remyelinate and produce myelin, actually can acquire some immune properties, and this is quite surprising and a new aspect that we are now very interested in follow-up. And other researchers in the US, for instance, Professor Peter Calabresi and Alban Gauthier, also have similar findings, and I think it's opening a new field to investigate what these progenitor cells can do.
Okay, so yes. there's a lot of groundwork that needs to happen first into that basic biology of remyelination that then eventually can be used towards developing some therapies in the future.
Precisely.
It sounds like a really exciting field and something new to add into that treatment landscape.
Yes.
So thank you so much to you and your team for all the hard work that you are doing over the years to contribute to some long-term improvements in our options. That's great. So a big thank you to you for coming along today as well. We appreciate your time.
Thank you, Brianna.
All right, so still to come, we've got a few more speakers. We have Professor Tanuja Chitinous, my apologies. She's travelled all the way from Boston in the United States, and she's going to be talking to us about integrating patients with research and self-reported outcomes in the field of MS. And then we're also going to speak to a Dr. Chris Dwyer, who's come all the way from here in Melbourne, Australia, and he's going to be telling us about the evolution of the MS treatment landscape in Australia and where to next. Before we get on to the next speakers though, I just wanted to mention a new fundraising campaign that the MS Research Australia team launched this year through Kiss Goodbye to MS, their national program of fundraising, and that's the May 50K that happened earlier this year. So the May 50K was a concept where it challenged participants to either run or walk 50 kilometres throughout the month of May to raise funds to leave MS where it belongs, which is behind us. So as one of the other 25,600 Australians who are living with MS currently, my motivation for taking part this year was kind of a twofold thing. Firstly, I actually used it as a tool to make my MS public knowledge. Prior to that point, I'd only really told a few select friends and family about my diagnosis, and I was ready to let more people know, but that's where my second motive came into being. I thought that by doing it in conjunction with the May 50K, I could actually have a bit of a tangible benefit by raising funds for MS research, and I felt like that was the perfect opportunity to do it. Turns out the collective efforts of the MS community in this fundraising campaign were incredible. We raised $2.1 million, which was far and away more than what the target had been, so that was a fantastic effort by everyone involved, and if some of you at home watching now did that as well, thank you so much. So for the May 50K campaign, here's a little bit more information about what it was. So the May 50K, I don't know about you at home, but I'm definitely signing up again for next year. It's vital that campaigns like this allow us to raise the funds that are really crucial to allow the kind of research that's we've heard about this morning, that research can continue. Because eventually it is these developments that happen with these researchers that eventually goes on to creating some treatments that will help us stop and reverse MS, which is what we're all aiming for. And yeah, May feels like it's a little while off. We've still got to get through Christmas first, but there's no time like the present to register. You can hop online and do it. So please register to take part in the event for next year. You can register as an individual. You can set up a team. Could even get your workplace involved if you felt like they might be willing to help you out as well. It takes less than 2 minutes to register, so please get online, themay50k.org. You can do it today as soon as the live stream's finished, of course. Now, I think I heard on the grapevine that you set your goal insanely high, 250 kilometres. Are you going to up the stakes next year?
That was a bit of a struggle, but it was great fun. But I've started training already, so why don't we have a bit of a competition? I'll match whatever you do.
Oh, it's going to be lower stakes, but we'll give it a go. Sounds good.
Yeah, but the whole team at MS Research Australia is really looking forward to the May 50K 2020. It was a great success this year. It raised lots of money, but it's also about getting out, being part of it, being active, and really having fun. So it was really good. Now up next we are hearing about integrating patients with research, self-reported outcomes from Professor Tanuja Chitinous. Professor Chitinous is based in Boston in the United States at the Harvard Medical School and the Anne Romney Center for Neurological Diseases at Brigham and Women's Hospital. Her work focuses on moving laboratory discoveries into clinical trials for MS and related disorders. She oversees a team of analysts and researchers working to try and understand the causes and differences in response to treatment in people with MS, and is investigating why MS progresses differently amongst diagnosed people to inform new treatment. She also has extensive experience in sex difference in autoimmune disorders and paediatric MS research. So welcome, Professor Chitinous.
Thank you.
So to start with, you've been involved in long-term studies following people with MS. Have these studies revealed anything to help with predicting disease outcome?
Well, thank you for asking that question. You know, as a clinician, I'm asked every day questions like, will my MS progress? Do I have a more severe or a more milder form of MS? When can a next relapse happen, and which is the best treatment for me? And so to that end, we've set up a study called the CLIMB study, which is called Comprehensive Longitudinal Investigations in MS, and our goal is to answer these questions through a number of different measures. We also know that MS is a complex disease, and so it's not one measure or one biomarker that's going to yield the answer to any of these questions, and we have to merge different data streams together in order to answer such questions. And so to your question, can we predict disease course? We are getting to that, and now with combining many different measures such as clinical measures, MRI features, volumetrics, blood biomarkers, genetics, we are now coming to algorithms, and machine learning algorithms are also helping us to identify who is going to have a more aggressive disease course, and in that way clinicians can maybe institute treatments earlier for those people.
Yeah, so kind of gathering kind of more information about each individual, I suppose, is really what the aim of the game is. So we often hear about self-reported outcomes. What are these, and how are they helping with research?
Yeah, so the goal of research is really to help people with MS live their daily lives. And in order to do this, we need to understand how they're feeling on a daily basis. And that includes measures like quality of life. How long are they working for? Fatigue, mood disorders, and to that end, we ask questions, which includes questionnaires, to address these questions. The information collected from these questionnaires can then be used to identify better treatments for people with MS.
So I suppose it's also important for clinical management, not just research as well. One subgroup of people with MS is children. It's often an area not talked about. But what percentage of people are first diagnosed as children, and is their MS any different to adult onset MS?
Yeah, so thank you for asking that question. And about 5% of all MS patients probably have their first symptom in childhood, and that includes childhood as well as their teenage years. We know that teenage or adolescent onset MS is more inflammatory, and typically these young people have higher relapse rates than the typical adult patient. So it does make it challenging to manage, of course, to live with an inflammatory form of MS, and we're just starting to see treatments for pediatric onset MS. And in fact, the first treatment for pediatric MS was approved around the world as well as in Australia in the past year or two.
Yes, that's recent news here in Australia as well, which we're very pleased about. So thank you very much for taking the time and those insights. We really appreciate you coming all the way to talk to us here, but also at our scientific conference. We've still got more to come. Now we've got our homegrown Australian MS expert, so there's a little bit more time to submit questions for our Q&A panel discussion coming up towards the end of this show. Simply send us a message at inquiries@msra.org.au. The email address should be on your screen now. Also, you can send us a message via Facebook. Remember, if there's people that you think might be interested in the stream, if you're watching via Facebook, you can share it, or you can forward it to your friends and family who may be interested. And of course, join the conversation by using the hashtag MSLiveUpdate19 to join the conversation online. Back to you, Roni.
Thank you, Hamish. So now it's time to hear from our final expert for today, who is Dr. Chris Dwyer. Dr. Dwyer actually works from the location that we're filming at today, which is the Florey Institute of Neuroscience and Mental Health. He's also a practicing neurologist at the Royal Melbourne Hospital. Now, Dr. Dwyer's research, it focuses on a particular gene called MERTK in the immune system, which has been implicated in the development of progressive MS in particular. And he's wanting to determine whether this gene MERTK acts via immune cells in the brain and spinal cord or via any kind of cells in the blood as well. He and his team's aim is to develop a treatment strategy eventually based on MERTK that will target the right type of immune cells in progressive MS, which is rather exciting. And today he's going to be speaking to us about the evolution of the Australian MS treatment landscape and where to next. So welcome, Dr. Chris Dwyer. Thank you for taking time out of your very busy day today. Look, I think the first probably important point to talk about is all the treatment options available for people living with MS. When I was diagnosed 3 years ago and you're rifling through all the fact sheets that you're given and seeing what's out there, one thing I noticed is that the number of treatment options available, it's really exponentially expanded over the last 10 or 15 years. So as a neurologist, how do you think this impacts your patients?
Well, I can say, first and foremost, it makes our consultations a lot longer. We have a lot to discuss. We're spoiled for choice, and that's really a reflection of the amazing work that's been done over the last 15 years, and so we're very grateful to be in that sort of luxury position. So I think that amount of choice, being able to choose between injectable therapy or oral therapy or infusional therapy, has really empowered patients and taken away some of that initial fear when they're first diagnosed that I'm sort of on my own here and I've got this condition that I've heard about, that we've actually got a lot of things to offer them. So there's a sort of psychosocial aspect to the importance of that development. But also what we know through the work of my colleague Thomas Glenchick at the Clinical Outcomes Research Unit at the Royal Melbourne Hospital is that having that number of highly effective therapies for relapsing remitting MS is delaying the time to which people will get progressive forms of the disease. So we've got pretty good evidence now that being on treatment early, being on highly effective treatment early, pushes out the time horizon for that. And that's a really important piece of information for our patients when they're deciding on which therapy to take.
Absolutely, and during the course of their condition, the fact that you're able to change things if one thing's not working, to have the options to go to others.
Absolutely, that there is a hierarchy that we can use, or you can have a conversation with patients about starting with highly effective therapy first. That's a detailed conversation, but yeah, there's always something else we can try at the moment.
Okay. And so, of course, there's always more and more that's coming through the pipeline. So what is it that you would say would be the most promising development in MS treatments at the moment?
Look, we do have a few interesting trials that are going to report in the relatively near term. There's a trial looking at high-dose vitamin B as an energy supplement for patients with progressive disease. We've been using that in a sort of off-label capacity, and there may be a suggestion that for at least a subset of patients that will help with some of the fatigue fatigue symptoms and brain fog symptoms that they describe. The first agent looking at remyelination will be reporting in the relatively near future, alpecinimab, so it'll be interesting to see if there's a clinical benefit for patients in taking an antibody that is specifically targeted at remyelination.
Okay.
I think at the moment there isn't a sort of breakthrough drug going to report on the horizon in the very near future. I think some of the really interesting developments that we have at the moment are more centered on the patient-centered care idea. In particular, we're all walking around with these sort of veritable supercomputers in our pockets these days. And the amount of data that you can capture with one of those is really quite extraordinary. That they've got accelerometers, you can do coordination tests with them, they can measure how many steps you take a day. And we use quite a coarse tool to assess patients and their clinical stage, this tool called the EDSS, which I'm sure many of your patients have heard of. It's not an ideal measurement.
It's not comprehensive.
It's not comprehensive. It's very heavily weighted towards walking.
Yep.
And so having more granular information about how our patients are going through the use of something, for example, as a smartwatch or smartphone, I think that's really got some interesting implications, not only for patients for how they're going, but for the big data we can collect from a large population.
Okay. Wow, so there's a few different kind of aspects of
yeah, it's not just drugs. It's not just drugs.
Good.
I think that's what we want to reinforce for people.
Yeah. In terms of drugs, though, of course, obviously relapsing, remitting are the ones that are benefiting most from all these developments, whereas progressive MS, historically there's been far fewer treatment options. So what can you tell us about how this might be changing?
Well, we have one agent that seems to show benefit for patients with primary progressive MS, at least a subset of those patients, likely in the setting of ongoing inflammatory activity. I think there's important information to be gathered in the course of the next couple of years looking at the use of that same agent in patients who don't have inflammatory activity. And so we're part of this study that's to initiate soon here in Melbourne, looking at the use of ocrelizumab in patients with really quite high levels of disability, wheelchair-bound patients. It's really quite a brave initiative on the part of the sponsor to put that study forward. Wheelchair-bound patients who obviously can't use an EDSS, so we have to use a novel endpoint for them, so looking at their ability at a 9-hole peg test. So I think that's an important trial for us to be able to distinguish which patients are going to benefit from a treatment that is not, must be said, without risks, and which patients won't get any benefit from that. And so I think that's a really important initiative that we've got going at the moment.
Okay, that sounds great. Now, I've gotten very sidetracked. I mentioned the research that you've been doing into the MRTK, so we should probably get back onto that.
Sure.
So can you tell us a little bit about the research you're doing into that?
First and foremost, I'm one of only many here at the Florey who are looking at MRTK as a potential target. We've heard earlier today that there's over 200 genes implicated in the development of MS. MRTK is one of those. It has to be said its contribution is not enormous, but it is a signal certainly from genome-wide association studies that MRTK is important. My work has been looking really in two aspects of MRTK. One in humans, looking at MRTK levels in the blood on specific immune cells from patients when they're having a relapse, and then getting them back a few months later when they're in remission to see if there's a change in the level of expression of that of the protein that gene encodes based on clinical status. Headline is it's probably not going to be a relevant change in the peripheral blood of patients, and that doesn't mean that it's not an important target, just that we can't capture it in peripheral blood. That may be because all those cells that are important have migrated to the central nervous system. I've also been working with mice that are deficient for that gene in a very special immune cell called a dendritic cell, and we have some interesting preliminary outcomes there that was presented at the MSRA conference this week. The male mice that are deficient for this gene suffer from a condition called EAE, which is a mouse model we use for MS. No other animal gets MS, so we have to do some tricks to their immune systems to make them get a disease that looks like MS. But the male mice that are deficient in this gene in their dendritic cells get a worse form of this condition. That's got some important echoes in the human condition, whereas in relapsing remitting disease, of course, it's just the population is skewed towards female patients. Progressive disease is not, it's much more equal. And among the male patients we know from MS base and studies done around the world that male progressive patients tend to do worse. And so there's interesting echoes, as I said, in the human condition there. So a lot more work to be done to unwind exactly why the males are doing worse, but the influence of this gene on their dendritic cells does seem to be having an effect.
Well, it's promising you've been able to kind of draw that parallel into that animal model, so that's
definitely
that's
warrants further investigation.
Yeah. Okay. And of course, research is the focus of all this, and you know what we need the crucial funds for. So, for you as not only a researcher but someone who's at the coalface with patients living with MS, what does your research, and also just research in general, what does that mean for people who are living with MS?
Look, I think my research in particular, I'll be very honest with you, it's far away from the sort of clinic or having a clinical application at the moment. And so when I'm recruiting my patients who very kindly give us blood when they're having relapses, and then when they're in remission, I have to say to them, this is probably not going to help you. This is for posterity. But research overall, of course, keeps patients engaged with the community, keeps patients engaged with their disease, and provides hope and a scientific community in which to experience your disease and move forward with support. And I think that's we know from long-standing evidence that being in a trial and being in a clinical scenario has secondary benefits for patients, not just the scientific information that we gather. So research is essential for driving this disease forward and keeping patients engaged and hopeful.
Yeah, and it's the big picture of what we could be aiming for in the future, to be developing those new strategies. And
That's right. This is a journey we're all on together.
Exactly.
Yeah.
Perfect. I think that sums it up very nicely, Chris. So thank you for that. So look, it was great to hear about your research as well.
Thank you for your interest.
Even though you say it's far from the clinical coalface, I think it's all working towards a common goal. It's every little piece that
we're certainly hopeful.
Puts the puzzle together. So I really hope that everyone at home, you've all enjoyed hearing all the information that our researchers and neurologists have been able to provide for us today. I know that I certainly feel like I've gleaned a few little extra hints and tips and ideas of what's going on behind the scenes. So now, speaking of behind the scenes, I know that the MS research team have been working away very hard collating all the questions that have been coming in from you at home, so that we can get our researchers' responses to those. So it's time to hand back to Hamish from MS Research Australia, who's going to be chairing this section.
Excellent. Thank you, Baroni. Yes, we have received a huge number of questions. Unfortunately, we're not going to have time to be able to answer all of them. Some of the questions did have very similar kind of themes, so we have tried to kind of compile some questions that may hopefully kind of address the questions that a lot of you do have. I should also note, unfortunately, we cannot give advice or clinical advice about treatments and more personal options like that. However, if you do have concerns about treatment, we do urge you to speak to your neurologist about any concerns you have. Now I'd like to welcome back our 4 MS experts and researchers. We, of course, have Alan and Tanuja, Gonzalo and Chris. So thank you very much. So we've got some questions coming in. So I might as well kick it off to start with the first question we have from Norman. And Gonzalo, this is probably kind of coming your way, a bit of a heads up. What are the new treatments for remyelination, and how long will treatments be available? How long will it take for these treatments to become available to the public?
So we addressed a little bit this question before. I mean, there are several clinical trials that have some promising results. One of them is, for instance, Lingo, an antibody against Lingo, which inhibits the process of remyelination, which had been 2 trials, one with positive results, one with not so positive results, and now there are strategies to try to redesign the trials. There is trials with drugs that have been used for any indications and that have been repurposed for clinical trials for MS. One example is clemastine, that also has in the first trial shown some positive results, but we are waiting for more results. So there is many trials ongoing, but one thing that I would like to emphasize is that we really need to to find new targets, do more fundamental research to find what part of the remyelination to trigger. For instance, some of the latest fundamental research has indicated that, for instance, the progenitor cells, the cells that give rise to oligodendrocytes that produce myelin, seem to be depleted or reduced with MS. And so we have to find new strategies to increase the number of these progenitor cells, and then so target both at early early stage, but also then the later stage when we have to produce new myelin. Another example is like these findings that we have had that progenitor cells might have some immune properties, and this also opens very exciting perspectives in the fact that most of the treatments currently in MS tackle the immune system, but they might also affect these progenitor cells that we are seeing now have immune properties. So I think that it's very promising, it's very exciting times because we are starting to find pathways that we might be able to tackle for remyelination.
Yeah, excellent. So I suppose, does any other panel want to add anything to that? I suppose we often talk about remyelination in progressive MS, but what about relapsing remitting?
Yeah, I think some of the data that came out of the conference over the past 2 days show that there may be opportunities even in the relapsing remitting phase to promote remyelination, perhaps at the time of an attack, where we see from the research that was presented that actually cleaning up the damage that's done at the time of an attack or the development of a new spot on the MRI scan may in fact kind of pave the way for improved remyelination. So there are great opportunities even early on to pave the way for recovery and repair.
Yes, and there are also some new opportunities, I think, with clinical trials, especially in progressive forms of MS, which might also be important in earlier MS. And here there are trials where mesenchymal stem cells are being transformed and injected back into the spinal canal with the hope that these will promote either repair or remyelination or help to just modulate what's going on in terms of damage in the CNS.
Yes, no, it's definitely an interesting area. So the next question we have from Terry, who has a progressive form of MS. I suppose it's probably very, very similar, but it is, will there be anything in the near future to help stop my progression? So I don't know who wants to go first.
Yes.
Chris, maybe.
Well, Terry, the short answer is we just don't know. To stop progression completely would require understanding the basic mechanisms of what drives progression, and there's a Nobel Prize for the person who cracks that nut. So we may have some medications that might be able to slow progression. I think that's the very best that the field can offer at the moment for a subset of patients who've probably still got inflammatory activity. So if you've had progressive disease for 10 years and you haven't had a change in your MRI for a very long time, you're probably not going to benefit from those medications, but that trial that I alluded to is going to help us determine that.
Excellent. Does anybody else have any thoughts on that?
Yes, I think it's very exciting. I mean, there are a number of new targets in several clinical trials which have been launched now in progressive MS, and I think strategies to try to help the nerve cells to work better, to provide more energy. There is a trial that will read out in the next year, which is very promising and exciting, and other strategies that are following along that to help the nerves just work better might be useful for patients with progressive MS.
Excellent. And I suppose we're hearing a lot about trials. I will just point out we do have a website, mstrials.org.au, where we do list all the clinical trials that are happening in Australia. So if you are interested, make sure you go to that website to have a look at the trials which are busily recruiting here in Australia. So anyway, moving on to the next question from Catherine. What are the panel's members' view on the importance of lifestyle? And this includes diet, meditation, exercise on disease progression. Who wants to go first?
I can talk about that a bit. So I actually think that lifestyle modification, although we may not be able to test it in randomized trials, makes a lot of sense within reason. For example, the data we saw at the conference demonstrated that a reasonably healthy diet, nothing extreme where you're having to eat 20 cups of kale a day and that's it, but just a modestly healthy diet with a little bit of meat added back in and some fish and things might actually be beneficial for preventing MS. Our studies also show that using those kinds of diets in general populations are overall good for our body's health. They reduce the risk of obesity-related complications like diabetes and high blood pressure. And we are increasingly recognizing that those comorbid illnesses can actually have a negative impact on the prognosis of a person with MS. So I generally talk to people about pursuing just a generally healthy diet. We know that exercise is neuroprotective. It protects those neurons in our brain. So whether or not it has any direct impacts on remyelination or other things, if we're protecting our neurons, we're doing great things. And as we heard in the conference yesterday as well, sleep is relatively important for the health of our brains as well. So that's sort of the third part of the lifestyle arm, making sure that we're getting a good night's sleep.
So did anybody else want to add to that?
Yeah, I think I just add to that exercise, and I think we now all know that we should exercise on a relatively regular basis. But with people with MS, it's important to exercise and identify the best form of exercise that works for you. And now there are strategies to do that, working with a physical therapist, working with your doctor, to identify a form of exercise that's safe but also effective.
We had a great presentation yesterday, actually, about some research in exercise, right? Instead of more traditional, 30-minute slogs, or even short bursts of exercise that may be more tolerable for people with MS, could have really meaningful impacts on function.
And we actually also heard that only 4% of the Australian population, the general population, actually follows the Australian healthy dietary guidelines. So it's probably something we could all probably take note from. Chris, I don't know whether you want to add anything more to that.
I just add that there's one lifestyle intervention that we absolutely have to reinforce for everyone, and that's a general one as well. Smoking is absolutely the worst single thing you can do for your health, and if you've got MS, it's the worst single thing you can do for your health on steroids. So you absolutely must stop smoking if you're a smoker and you have this condition. But I think it's really important not to get too obsessive about the diets that we follow, and so I advise my patients, I don't want you remembering your MS with every morsel that you put in your mouth, because I don't think that's a fun way to live your life. And so just adopting sensible general principles for healthy diet and exercise is important. We know that protects neurons, as Professor Maury was saying, and your patients with MS have a condition where neurons are being affected. And so preserving what you've got for all of us is important, but more so when you've got an accelerated process going on.
Excellent. Well, thanks for that. So now I've got a bit of a curly question that's kind of come through from Natalie. Why do symptoms and disease progression rates vary so much between individuals? I don't know who wants to start with that.
Yeah, that is a complex question, and I think we're going to start to get to it through these large longitudinal studies, which are being conducted all around the world, but including in Australia. There are several efforts which have now started. But it probably is a combination of background genetics, it's probably environmental factors, and just features of the disease course might predict how you do. So we know that there are some people who have a more aggressive form of MS. They might accrue disability faster. There are some people who have milder forms of MS, and it would be very useful to identify what's driving that, maybe give them a better or more aggressive treatment earlier on. And so I think the short answer is that we're working on it, but there is more and more information, and we might be able to now predict disease course with some of these measures, biomarkers, and algorithms.
Yeah. Anybody else want to add to that?
I just think it's important to remember this disease is the intersection of the body's two most complicated systems. So your immune system and your nervous system are both fiendishly complicated, and when one is not playing well with the other, that makes it very difficult for us to tease out the disease mechanism and to have hard and fast opinions or rules about how people are going to fare with this condition. And so each patient's MS is unique to them because their immune system is unique to them, and the environmental and lifestyle exposures that they've had are unique to them. I think that's important to bear in mind.
Excellent. Yeah, I think that's probably a really good kind of summary of the situation. So the next question is actually probably very similar. Is research any nearer to telling us our prognosis? Are we nearly at the point where we can say this individual will have this outcome? And that was from Suzanne.
Yeah, I think that being at this conference for the past couple of days has highlighted that there are large research initiatives going on trying to capture data more broadly about people, and then facilitating the capacity to put those data sets together so we really can start to identify subgroups of people who may be at higher risk for certain types of disease courses, particularly progressive MS, compared to those who may not be. And it's really that kind of collaborative spirit that I think is developing worldwide that's going to help us to identify the subgroups and then to be able to go back at the individual level and say, well, you fit into this subgroup. That's probably what your disease course is going to look like, and to think about whether or not we can approach that person with a different kind of treatment strategy in mind compared to somebody who may be in a different bin right from the start.
Yeah, and I suppose that also helps reduce some of the fear that may have been, or the fear of the unknown of what it could be. And I suppose the other thing I'll say, it's actually quite exciting to that a lot of this research is actually happening here in Australia. So I think it's quite an exciting area. Does anybody else want to kind of add to that, or? No, I think it probably sums it up kind of quite nicely. We probably are running out of time, so that's probably all we have time for. So once again, I'd like to thank the panel for your contribution and your insights, and we really appreciate taking the time to talk to us today. I'd also like to thank our major sponsors for the event, Roche, and also the Fleury Institute for Neuroscience and Mental Health for allowing us to use this venue so we can stream to you today. And I'd also like to thank my fantastic co-host, Bryony, for all her hard work.
Oh, look, there's a lot of people behind the scenes that are doing the hard work. I'm just sharing the stage with you, and it's been my pleasure to do so. So thanks, Hamish. Now, for you at home, if you have enjoyed our live stream, but you feel like you might have missed something, or there's something you just wanted to watch again just to really get it in, you're in luck. We are going to post the video online so you can watch again at your leisure. Likewise, if you've watched it and thought, oh, gee, my friends or my family, I'd really like them to see this as well, as I say, you'll be able to find it online, so please do share the on-demand version of the event. It will be available to watch. You can see our website on the screen there, so msra.org.au. It will be uploaded shortly.
Yes, and it also will be on our Facebook, and also the link will be on our Twitter feed as well. But to get the latest MS research news directly into your inbox, make sure you go to our website, msra.org.au, and sign up to your newsletter, and we'll send out regular updates of what's going on. Also, don't forget that this incredible MS research being done here in Australia can't be done without your support. So please do go to msra.org.au/donate to donate. There's a big orange button, and we would really appreciate your help in helping us stop and reverse MS. Finally, I'd like to thank everybody at home for watching this live stream, and I hope you have a good rest of your day.